It's 1958. Two scientists, Matthew Moselson and Franklin Stahl, are trying to solve the greatest mystery in the history of science. And that's, how does life copy itself? And so Watson and Crick had discovered the double helix 5 years ago, but nobody knew how the DNA actually replicated without destroying itself. Moselson and Stahl decide to run an experiment. They grow bacteria in a heavy nitroblip and switch them to light nitrogen. They spin the DNA in a centrifuge for 48 hours. When they look at the results, they don't just see a band of DNA. They see the exact mechanism of immortality. They prove that DNA unzips down the middle, and each half builds a perfect new partner. It's called semi-conservative replication. Biologists call it the most beautiful experiment in biology.
Why are we telling you this? Because if you understand how Moselson and Stahl designed that experiment, you don't just understand DNA. You understand exactly how the College Board is going to test you. You see, every year, over 280,000 students take the AP Biology exam. And every year, only about 18% of them get 5. They fail because they treat AP Biology like a vocab test. They spend hundreds of hours memorizing the steps of the Krebs cycle, and then they walk into the exam and they realize the exam doesn't care if you memorize the Krebs cycle.
The College Board cares if you can look at a graph of an enzyme denaturing an acid and predict what happens to the cell. AP Biology is not a memorization test. It's a data interpretation test disguised as a science exam.
I'm Sovik. I got a 1580 SAT and full ride scholarships into my dream schools.
And I'm Zach. We are Ivy League mentors at Ignite Test Prep. Today, we're going to do something no other AP Bio video has ever done. We aren't going to give you a list of organelles to memorize. We're going to show you the actual source code.
We're going to break down for you the exact scoring algorithms that College Board uses. We're going to reveal the big 4 ideas that connect all 8 units. And we're going to teach you the exact formula to hack the 9-point experimental design.
This is the only video you're ever going to need to watch to get a 5 on AP Biology. So let's start it.
Now, before we teach you the biology, we have to teach you the game. You can't win a game if you don't know how the points are scored. The AP Bio exam is graded on a raw composite score of 120 points. To get a 5, you don't need to be perfect, but you do need roughly 85 to 90 out of the 120 points. That's about a 75%.
And here's exactly where these points are going to come from. So in the first section, section 1, it's going to be multiple choice, 60 questions, 90 minutes. And it's going to be worth 50% of your score. But there's a catch, because almost every single question is stimulus based. And so they're going to give you a paragraph about a mutated fruit fly, a data table, and a graph, and ask you 4 questions about it.
Rule number 1 of the AP Bio MCQ, do not overthink the data. When you look at a graph, College Board isn't testing if you can read the exact number on the y-axis. They're testing if you can recognize a trend. Is it going up? Is it going down? Does it level off? You have to find the trend and connect it to a core biological principle. If the graph shows enzyme activity dropping to 0 at high temperature, the answer isn't about the specific temperature. The answer is the protein denaturing.
In section 2, this is the free response questions. So the FRQs, it's going to be 6 questions, and they give you 90 minutes. So it's also worth 50% of your score. So there's going to be the 2 long FRQs worth 9 points each, and 4 short FRQs worth 4 points each. And so the first question is always interpreting and evaluating experimental results. The second question is always experimental design.
Listen to me very carefully. You cannot get a 5 without dominating question 1 and question 2. They're worth 18 points combined. That's 15% of your entire exam score, wrapped up in just 2 questions. We're going to teach you exactly how to hack the rubric for these questions later in the video. But first, you have to understand the content way. Overall, there are 8 units in AP Biology, but they're not created equal. Unit 1, Chemistry of Life. Unit 8, Ecology. These are the 2 easiest units, which means College Board tests them the least. They make up maybe 10 to 15% of the exam combined.
The absolute meat grinder of the exam, basically. The highest yield units that will make or break your score are units 3, 6, and 7. And then we have cellular energetics, gene expression, natural selection. Those are the 3. And these 3 units alone can make up over 45% of your multiple choice questions, and almost always feature heavily in the long FRQs.
But here's the secret. You cannot study these units in isolation. The College Board doesn't ask questions about just one unit. They always ask crossover questions. They'll ask you how a mutation in DNA, which comes from unit 6, changes the shape of an enzyme, which comes from unit 1, which stops cellular respiration, unit 3, which causes an organism to die before it can reproduce, altering the allele frequency of the population, unit 7.
And to answer those questions, you're just going to have to understand the 4 big ideas that act as the invisible string tying the entire curriculum together.
The College Board literally tells you how they design the exam. They build every question around the 4 big ideas. If you understand these 4 concepts, you can logic your way out of any question, even if you forgot the vocabulary.
Big idea 1 is going to be evolution. The process of evolution drives the diversity and unity of life. Charles Darwin goes to the Galapagos Islands and looks at finches. He realizes that the birds with beaks perfectly shaped for cracking hard seeds survive droughts, while the others die.
Evolution is the why of biology. Why do mitochondria have their own DNA? Because billions of years ago, they were independent bacteria that got swallowed up by a larger cell, the endosymbiotic theory. Why do all living things use ATP for energy? Well, it's because we evolved from a single common ancestor that used ATP. If a question asks you why a structure exists, the answer is almost always because it provided an evolutionary advantage that increased survival and reproduction.
And now we're going to move on to big idea 2, which is energetics. So this is just biological systems use energy and molecular building blocks to grow, reproduce, and maintain dynamic homeostasis.
Now, unit 3, photosynthesis and cellular respiration. Don't memorize every enzyme in the Calvin cycle. You want to memorize the flow of energy. Sunlight hits a leaf. The chloroplast uses that light to rip electrons off of water, creating oxygen, and then it uses those electrons to build a proton gradient. That proton gradient powers a molecular turbine called ATP synthase to make ATP.
Then cellular respiration does the exact opposite. It takes glucose, rips the electrons off, builds a proton gradient in the mitochondria, and makes ATP. It is all about the protein gradients. So if the College Board gives you a poison that makes the mitochondrial membrane leaky, what's going to happen? The proton gradient just disappears. ATP production stops, the cell dies. That is a guaranteed 2-point FRQ question.
Big idea number 3 is information. Living systems store, retrieve, transmit, and respond to information essential to life processes.
This is the genetics and cell communication. DNA is the hard drive. RNA is the messenger. Proteins are the machines. The central dogma of biology.
It's also about how cells talk to each other. A hormone locks into a receptor on the outside of a cell. That receptor changes shape. It triggers a cascade of chemical reactions inside the cell, a signal transduction pathway that eventually reaches the nucleus and turns a specific gene on or off.
Here's the classic AP Bio trap. They will give you a massive, terrifying diagram of a 15-step signal transduction pathway with names that you've never seen before, like MEK and ERK and MAPK. But I want you to know you don't have to panic. Just follow the arrows. If the question says protein X is mutated and cannot be activated, put your finger on protein X, cross it out, and everything downstream of it just stops.
That big idea number 4 is systems interactions. Biological systems interact, and these systems and their interactions exhibit complex properties.
So this is where ecology meets biochemistry. How does a change in the pH of the ocean denature the enzymes in coral, preventing them from building reefs, which destroys the habitat for fish, creating or just crashing the entire food web?
You see, everything is connected. A change at the molecular level cascades up to the cellular level, the organism level, and even the ecosystem level. If you can trace that cascade, you're thinking like an AP.
Now we get to the most important part of the exam, the FRQ question 2, experimental
Thank you, Ignite Test Prep.
design. It's worth 9 points, and you're given a scenario and asked to design an experiment or analyze an experiment. So here's the exact formula you're going to use to get all 9 points.
Point 1, identify the independent and dependent variables. The independent variable is what you change. I change the I independent variable. The dependent variable is what you measure. This is the one that's dependent on the independent variable. Pro tip, the dependent variable must be measurable. Do not say I will measure plant growth. Say I will measure the height of the plant in centimeters after 14 days.
And then point 2 is going to be identify the control group. So every experiment needs a baseline to compare against. If you're testing a new drug, the control group gets a placebo. If you're testing how temperature affects an enzyme, the control group is the enzyme at its normal optimal temperature. And I'm going to give you something else, and we call this the rubric hack. So always state why you have a control group, because the control group, well, let's say you have a quote that says the control group is used to ensure that any changes in the dependent variable are solely due to the manipulation of the independent variable.
Point 3, identify the constants, the controlled variables. These are the things that you keep exactly the same across all groups so they don't mess up your data. Temperature, pH, light intensity, volume of liquid. You have to list at least 3.
And then for point 4, you're going to have to state your hypothesis. So do not write a vague guess. You have to use the if, then, because format. And so a bad hypothesis example would be the enzyme will work better hot. But a good one would be like this. If the temperature of the reaction is increased to 40 degrees Celsius, then the rate of oxygen production will increase because the molecules will have more kinetic energy, leading to more frequent collisions between the enzyme and substrate.
Points 5 through 7, graphing the data. They're going to give you a data table and ask you to graph it. This is free money. But students lose points here constantly just because of stupid mistakes. First thing is the title. You have to include both the independent and dependent variables. Second thing is axes. The independent variable always goes on the x-axis. The dependent variable always goes on the y-axis. Third thing is labels and units. Label both axes and include the units in parentheses, like time in minutes or concentration in molarity. Fourth is scaling. Your numbers have to be evenly spaced. Don't just copy the numbers from the data table onto the axis if they skip around.
And then point 8 is going to be the error bars. And so they're going to give you the standard error of the mean, or basically the 95% confidence intervals. So you're just gonna have to draw the error bars on the graph. And the golden rule for this is gonna be if the bars of the 2 data points overlap, the difference between them is not significant. The golden rule of the error bars is gonna be if the error bars of the 2 data points overlap, the difference between them is not statistically significant. And so if they don't overlap, the difference is significantly. If they do not overlap, the difference is statistically significant. And so you're gonna write that exact phrase on the exam.
And point 9, predict and justify. They're going to ask you to predict what would happen if they change the experiment. What if a mutation changed the shape of an active site? You have to say your prediction very clearly. The reaction rate will drop to 0. Then you have to justify it with biology because the substrate will no longer be able to bind to the altered active site, preventing the catalysis of the reaction.
So follow that exact structure, use that exact vocabulary, and you're gonna walk out of the hardest question on the exam with 9 out of 9 points.
Now it's time to address the biggest trap in AP biology, the vocabulary illusion.
So your textbook is about 1,200 pages long. It contains over 3,000 bolded vocab words. If you try to make flashcards for all of them, you're just gonna drown. Let's be honest.
Exactly. The College Board, it doesn't test for trivia anymore. They redesigned the exam specifically to stop teachers from just forcing their kids to memorize the textbook.
So when you see a terrifying word on the exam, like the phosphofructokinase, do not panic. If you don't remember exactly what it does, look at the prefix and the suffix. This is going to tell you so much. So the phospho means it involves a phosphate group. And then the kinase, it's an enzyme that moves phosphate groups around to turn things on or off. You don't need to know the exact step of the glycolysis it belongs to, but you just need to know it's an enzyme acting as a switch.
And one quick tip is just focus on the verbs, not the nouns. Understand what denature, catalyze, inhibit, transcribe, express, select. Understand what these verbs mean. If you understand the action, you can figure out the actors based on the context of the question.
Exactly. And so now we're going to tell you our Ignite blueprint. So getting a 5 on this AP biology exam, it's not about having a photographic memory, and it's not about who read the Campbell biology textbook the most times. It's actually about who understands the architecture of the exam.
At Ignite Test Prep, we don't just teach you the facts. We teach you the algorithms, the cheat codes. We teach you how to look at a terrifying 15-step signal transduction pathway and instantly spot the one protein that matters. We teach you how to look at an impossible exam and see the matrix.
And that's why our students just don't pass. They dominate. It's why we have a 100% success rate placing our students into Ivy League universities. We engineer academic outcomes that other consultants think are impossible.
So if you're tired of the guesswork, if you're tired of studying for hours and still getting a 3, if you want a way to guarantee your results, whether it's a 5 on AP Bio, a 1550 plus on the SAT, or an acceptance letter from Stanford or Harvard, you need elite mentorship.
So click the link in the description. Go to ignitetestprep.com, book a consultation with us, stop playing the game blindly, and let's give you the blueprint to win.
Study smarter, not harder. Master the rubric, and we'll see you at the top. Catch you on the next one.